Hello, this is Jim Schnabel giving talk number 25 in a series from the 7th edition of Principles of Biochemistry by Albert Lehninger. Today we'll be discussing fatty acids and vitamin B12 and go through this rather interesting topic. Fatty acids provide 80% of the energy needs in the heart and the liver. They are broken down to acetyl-CoA, which goes into the citric acid cycle. The process is called beta oxidation, where the hydrophobic fatty acids require transport molecules to facilitate passage. The sources of fat include diet, cell storage. They can be synthesized, or you can have autophagy of fatty acids. They have these functions. They're an energy source in hibernating animals and migrating birds. There's absorption of fatty acids in the small intestine. Micelles are formed from combination with bile salts, and these are synthesized from cholesterol. Lipases convert the lipids to mono and diglycerides, and they're packed with cholesterol and proteins into chylomicrons. Apolipoproteins are proteins which combine with lipids and cholesterol, and they form lipoproteins. The lipoproteins can be centrifugally separated by component density, and these have different functions. So you get high density lipoprotein, low density lipoprotein, very low density lipoprotein, chylomicrons in the supernate at the top. You sequentially lose triacylglycerols, which increase cholesterol and protein fraction as you progress through the sequence of the lipoproteins from very low density to high density, or so-called LDL. Neutral lipids, coated perilipins, are transported into the blood-bound albumin, and these are carried to multiple sites. They can bind 10 fatty acids per albumin molecule. Glycerol kinase converts glycerol to glycerol 3-phosphate, then converts glycerol 3-phosphate to dihydroxyacetone, and then to glyceraldehyde 3-phosphate. This is a sequence of this reaction. There's glycerol here. Glycerol kinase produces a phosphate ester, which is L-glycerol 3-phosphate. You then get a oxidation of the number 2 carbon with NADH forming. You get a carbonyl group here. This is dihydroxyacetone phosphate. And triphosphate isomerase gives the formation of glyceraldehyde 3-phosphate. You get oxidation here and reduction here. And then this entity goes into glycolysis. This is an example of, in the mitochondria, which is a site of fatty acid activation, acetyl-CoA synthase transfers the CoA to fatty acid. So you have ATP here, and the fatty acid CoAl synthase. This is the OR moiety, which can be of any length. And the carboxylate ion is adenylated by ATP and forms a fatty acyl adenylate and pyrophosphate. The pyrophosphate is immediately hydrolyzed to 2 molecules of phosphate ion. So this attaches here. You get this ester, CoA, then attaches here. So you need this energy linkage in order to drive this reaction to occur. AMP is released. The thiol group of CoA, the sulfur here, attacks the acyl adenylate, which is a mixed anhydride, meaning you've got carbon here and phosphate here, and these, as I've stipulated earlier, are high energy bond linkages. So AMP is displaced, and you get the thioester fatty acid here, which is fatty acyl-CoA. The energy is minus 15 kilojoules for the 2-step process there and there. And pyrophosphate is liberated, and that is energy of spontaneous reaction minus 19 kilojoules. Fatty acid oxidation has 3 stages. There's beta oxidation, the citric acid cycle, and respiratory chain transfer. There are 4 steps to saturated acid beta oxidation. The C2 and C3 double bonds are produced. There's hydration of the double bond, where C3 has a carboxyl group. C3 is then dehydrogenated, and you have acetyl-CoA formed and removed. That's what the English shows. This is what occurs in biological system. The R constituent can be of any particular length. You have this moiety here attached to the CoA from the previous step. We have a reduction of FAD with acyl-CoA dehydrogenase. So you get loss of a proton at this point with this intermediate here. The FADH goes into the respiratory chain. This hydratase, you have water occurring here, and you get bond linkage of proton added back to this, along with hydration of this linkage. So this you can see that there's 3 bonds here, 3 bonds there, and this is an unstable intermediate here, which hydrates immediately with the hydration occurring over this step. NADH is formed, and that goes into the respiratory chain, and this is where the oxidation occurs at this carbon. And this is a setup where CoA can be go into this reaction. You get liberation of acetyl-CoA, and you get the sulfhydryl link attaching at this point here. So the unstable intermediate with the double bonding that occurs at this juncture here helps drive the subsequent reactions, the reduction here at this point, and the cleavage from here to acetyl-CoA. And by doing that, you can eliminate sequential 2 carbon units, 14 to 12 to 10, and so on, where you end up with acetyl-CoA for the even numbered carbons. So there are 4 steps which are repeated to yield acetyl-CoA and ATP. Each pass produces FADH and NADH, which gives you 1.5 and 2.5 ATPs, and NADH. So that's the number of FADHs, and NADH is 1 less than the fatty acid length divided by 2. So that's how you can calculate from any given length of a fatty chain how many FADs and NADHs you get. The acetyl-CoA is oxidized to citric acid cycle. Again, 10 ATPs are produced per acetyl-CoA. And again, this is the watchword, 1.5 per FADH, 2.5 ATPs from each NADH. So if you have a 16 carbon palmitoyl-CoA, that gives you 8 acetyl-CoAs and 28 ATPs, and each acetyl-CoA produces 10 ATPs. So you have 8 acetyl-CoAs producing 80 ATPs. So the 16 carbons, you can do the mathematics from this point and determine using these numbers how many ATPs are going to be generated per X even length of carbon. So 80 ATPs from acetyl-CoA, 28 from the electron chain, you get 108 ATPs. And unsaturated fatty acids use isomerases and reductases to ultimately saturate and decarboxylate fatty acids. So you can use unsaturated fatty acids to get this formulation. So here's an example of one of these issues. This is linolyl-CoA right here. This is an 18 carbon unit. There's a double bond here at 9 and at 12. This is the chemical name of that. Beta oxidation gives you 3 cycles where there's acetyl-CoA. You come up with this moiety here with the double bond here. There is isomerase here, which ultimately creates a saturation here, but not there. You get another oxidation, which gives you 1 cycle, and to begin the oxidation of the 2nd cycle. Then there's a reductase, which eliminates the next double bond. You get oxidation with of this, with the reduction of this entity here, and then you get an enolYl-CoA isomerase here, and then you can get the structure to go into a form where you can do beta oxidation and get the remaining 5 acetyl-CoAs. This is one example of how you can eliminate and get isomerization to eliminate the trans linkages and or eliminate the cis linkage and get trans linkages in order to affect the remaining part of this synthesis. If you have an odd-numbered fatty acid, 15, 17, 9, you convert the propionyl-CoA to D-methyl-malonyl-CoA, and biotin, as we had seen earlier, is used as carboxylate propionyl. So here's propionyl, which is a 3-carbon unit. With the biotin and a propionyl carboxylase, analogous to what we saw in the citric acid cycle with ATP utilized, you get a methyl-malonyl-CoA right here. So now you've got 4, and then the epimerase creates an adduct to the sulfhydryl group into this carbonyl right there. And then you can get a cleavage with the mutase and obtain succinyl-CoA from this moiety here, where you're effectively trading these constituent groups, the proton and the moiety, the carbonyl and the CoA attached to it. Vitamin B12 has a confounding mechanism of reaction. There's a Corin ring system that occurs with cobalt, exists as a covalent bond with the deoxyadenylyl group. There's a free radical produced and it's rearranged, and you get a migrating group such as a methyl-malonyl group. This is vitamin B12 here. So there's a 5' deoxyadenosine here, the Corin ring with cobalt being covalently bonded and coordinate here to nitrogen and coordination bonds to 3 additional nitrogens. And below that, you've got an aminopropanol side chain hanging off of this ring here, and a dimethyl benzimidazole ribonucleotide here. So this is a Corin ring right here. The Corin ring system is this part of it, and there's an amino isopropanol at this point here. There are regulation transcription factors. You activate genes which produce proteins used in fatty acid oxidation. They also act to convert heart energy source from glucose in utero, the fatty acids in the neonate. Peroxisomes are plant organelles where beta oxidation occurs, and the difference is that the first reaction, the peroxisomes utilizes hydrogen peroxide, which is cleaved immediately by catalase to water and oxygen. They reduce the C2 bond to an alkene linkage, and which facilitates subsequent steps. This process is more efficient with long-chain fatty acids, and bacteria utilize a different array of beta oxidation enzymes. Omega oxidation is omega oxidation which occurs at the carbon opposite from the carbonyl carbon at the opposite end of the fatty acid, which you see in some animal species. So the terminal carbon is oxidized, and you cleave it to acetyl-CoA from the opposite end of the carboxyl group that we have previously assessed. Finally, there are ketone bodies. Acetyl-CoA is converted to acetone acetoacetate and beta-hydroxybutyrate. These are ketone bodies, these three components. The latter two cross the blood-brain barrier as an energy source to the brain during starvation. You also see keto bodies used as fuel in untreated diabetes mellitus and starvation, which allow for continued oxidation of fatty acids with sparing of acetyl-CoA. These are produced in the liver, but they're not consumed in the liver. And that will be it for our talk. Thank you.